And good morning, everybody. We're live. Congratulations. You have made it through the week. Another amazing week of food safety, food quality, all that good stuff that goes on all of our tables every single day without a second thought. But Doug and I know that that is being put there, and you know it's safe because of people who are behind the scenes making that way. And often it's an unappreciated thing, but we're here to say thank you very much. We appreciate all that you do. If you're new, pull up a chair. My name is Brian Armentrout with the Food Leadership Group, and every week at Friday at 8 a.m. Mountain Time, 10 a.m. Eastern, we do a live stream on LinkedIn and YouTube, and we talk about all things food safety. And again, this is a talk. This is not a presentation. This is not meant to be a lecture with PowerPoints and all kinds of fun stuff that are guaranteed to put you to sleep. This is meant to be an interaction. And our primary goal here is to exchange information. And I'm always really happy because our guest here today, Dr. Doug Marshall, is one of the best experts out there for conveying information. And Doug is the Chief Scientific Officer with Eurofins. And so, Doug, how are you doing today, sir?
Good morning, Brian. Always a pleasure to be with you and everyone else who invests their time with us today.
And what's so cool about Doug, and I'll embarrass you a little bit here, is unlike other people with the doctor and the experience and the pedigree before his name, Doug is one of those guys who's actually out there in the field doing stuff. And he takes the knowledge that he has and actually applies it to what people need to actually execute and manage food safety. And that's really a huge thing. And I know you're just an absolute road warrior, Doug. And I at least wanted to publicly thank you for that and all that you do for food safety.
Well, I appreciate it, Brian. Thanks.
And so Catherine is here. Good morning. So one of the things that we love to do here on the chat is we put in where in the world you're at. And Doug and I are pretty much neighbors. I'm in Loveland, Colorado, where it looks like day 3 of wind up here in Colorado. And it looks like you're at home as well, Doug.
Yeah, I am.
Nice. So let's have people put in here where you're at in the world. And we'll get kind of started here. So as we begin, everybody, please join us for a drink of your favorite beverage. Catherine's in Fort Worth. Nice. Cheers. Nice. Got my normal coffee going there. Hey, Sean, good to see you. Belmont. Oh, in Canada. Yes, that is correct. So one of the things here, Doug, that we love to do on the chat is torture the guests with the dad joke of the week. Are you ready? Are you ready for the dad joke?
I really don't have a choice, Brian.
No, you do not. So, Doug, how do you talk with a bacteria?
Scream at the top of your lungs.
Oh, no, it's actually very simple. You call them on their cell phone. Cell phone. Oh, Sean, you got it. Nice. Okay, good. And if you know, astute guests here with the chat as well, Doug, will notice that I have here in my background a new thing here. So this is, we have entered as of, was it last week or the 17th, somewhere around in there, the new Chinese lunar year. So we're now in the year of the horse. And so I've got the cool little horse dude back there. Had a grandniece and some family members visiting, and that was my grandniece's project. He put that together for me. So you've got the 4 different seasonal horses. This one happens to be the fire horse for this year. So there'll be a quiz at the end of the chat here today. Good to see Tim. So, man, we got a ton here, Doug, to go over. So I'm really ready to dive in here on this topic because this is something I think in food safety, and we were kind of, as we were getting here on the chat here before, we were talking about this and all the different parallels that are out there in the world relative to this. But really, it's one of these things, and I'm sure you see it when you're out in the field quite a bit, and when you're working with the Refrigerated Foods Association and all these different trade associations out there as well, is that we're only as good as the tests that we have. And also interpreting that information and applying it correctly. And so as we were talking here, this definitely applies in your personal life as well. So a great example of this is health, isn't it?
Yeah, absolutely. And the analogy I'd like to start us off with is very contemporary to me. Yesterday, I had my annual checkup with my cardiologist. prior to that, I had a battery of blood tests done. Some of these are quantitative indicator tests. Some of these are qualitative, presence or absence. And the results of those tests were discussed during my conversation with the physician. And in every year, it's, okay, how are my blood lipids? How's my liver function? How's my kidney function? And so forth. And the course of the next year then is dictated by what these test results reveal. During this, you're fortunate to get 15 minutes face-to-face with a physician now. And that's supposed to tell you how you are. For most people, when you wake up in the morning, the indicator test that you first perform is you look at yourself in the mirror and you go, wow, this is going to be a fantastic day. Despite the fact you look like you got run over by a truck, that's what you're basing what your day is going to be like. And then for those of us in food safety, we do a lot of validation testing to make sure that our processes are designed well and that they actually perform as expected. And then once we're using these validated controls, we need some routine verification testing to make sure that the validated control actually worked on the day we used it. This could be process controls, it could be sanitation controls, could be GMP controls, could be supplier verification controls. So in the absence of those data, basically what you are doing before you start manufacturing your product is, this is your preventive control verification test.
Yeah.
So I don't know, if you're a believer in that, fantastic, good luck in your career. But you really do need to be using these data to inform you on the health of your manufacturing process.
Yeah. And on the health analogy side, men do this. I know I'm really guilty of this, right? Doug, I've been fine. I've been healthy as a horse for all these years. Everything is fine. Sure, I smoke 7 packs a day and drink a bottle of Jack Daniels, but no, I'm perfectly good. And then all of a sudden, one day, the wake-up call comes and something happens. And all of a sudden, people change. And it really kind of parallels what we do in food safety if we're not paying attention. That base reality is there, whether we like it or not. And eventually, that recall is going to happen. That serious health issue is going to happen. There's going to be something that happens. And you'll see this in the real world in our personal lives. Somebody has a serious health event and all of a sudden they become this mega health person, right? And they only eat the right things and they're checking their blood pressure every day and their salt and you name it. And they're actually now managing it correctly. But it always comes back to, and this is always the leper's bell that I look out for in plants when I would visit, is if someone says to me, well, we never had a problem before, everything's fine, kind of extrapolating that past luck onto future results.
Yeah, there was a famous quote from a philosopher. His name is Neil Young. And the quote is, it's better to burn out than it is to fade away. Mm-hmm. And so there's a couple ways you can interpret that, just in terms of personal lifestyle, something to be said about that. Do you want to lead a hedonistic lifestyle and enjoy whatever minutes you have on this planet? With the risk of potentially dying young? Or do you want to live such a cloistered, sheltered life that you have no fun in life, but by golly, you're taking care of your personal affairs and you're going to live to 100? So the analogy I would kind of twist that for food safety is, the burnout part is you're constantly chasing problems. Mm-hmm. And as a result of that, you never get ahead of the game. And so it's just constant molecular motion in your brain and your body and with your teams trying to keep up with all the issues you have in a facility. Versus fading away is you're taking all of these data points that you're getting and you're doing proactive management to prevent all of this crap from happening. And that allows you to fade away with a long career, whether it's with your current employer or whether you move up And so that data is your friend, it's not your enemy.
Right. And that's a great analogy. And it's something I've been talking about on the chat quite a bit is, do you want to manage things reactively or proactively? And our health analogy is perfect for that, is figuring things out. And part of this as well is not going down rabbit holes, because you'll see that as well too. Some people just take it way, way too far, and they'll get too far down in the weeds. Or they will bring up anecdotal stories of, well, I know a guy who was a triathlete who dropped dead at 50. Oh, okay. Well, yeah, everything in life is a bell curve. And you have to look at that data and figure that out and look at the averages and the median and all these other fun things to kind of determine what that correct course is overall. On the side of the health analogy again as well, we were talking about blood tests too earlier. And when they say, they set up ranges for everything, and they say, all right, well, you're within the typical range for things like this. They say, okay, cool. And they use that data then to determine whether it's, thumbs up, thumbs down. Hey, Doug, we got to put you on a medication. Oh, man. And trying to figure those things out. But behind the scenes, as we were talking about earlier, is how are those ranges determined? Because a lot of times, too, you'll hear where like the American Medical Association will come in. I think recently, didn't they changed the limitations on your blood pressure for your systolic and your diastolic. Why? Why did they do that other than just moving it down? Or BMIs, you'll see this quite a bit too, where they're always monkeying around with the mass, your mass indexes associated with weight to height. What is the data set behind that? And then also figuring out, okay, where do I fit into that general data set? And then taking action around that. So I think the analogy around this with what we do in food safety is there is just an avalanche of data that's thrown at us, and trying to figure out how to move forward with that is a real challenge.
Yeah, I think that the other issue is the headbutt between what industry wants. So they want cheap and fast.
Right.
Okay. That's their reality. Versus the test kit manufacturers, the laboratories, the do-it-yourselfers, they're trying to deal with the ambush that industry gives them versus what the technology can actually support. Yeah. So if you want faster, you typically are giving up something related to the data quality. So it's either not sensitive, so you miss a lot of important attributes you want to find, let's say a pathogen test or an allergen test, because the limit of detection is greater than a slower test or a more expensive test. And so there's always going to be that trade-off between meeting what industry wants versus what the science can deliver.
Mm-hmm. And I would always, when I'm looking at laboratory testing, I would put it into two main buckets. So you got your chemistry tests and then your microbiological tests. And a lot of times in operations, people outside of kind of the laboratory setting confuse those two and think they're the same. It's like, well, if you can do this accuracy on this chemistry test, why can't you do it on the micro test? And it takes a lot of education. And I would always kind of distill it down to bacteria don't care. They will grow if they want to grow. And just because you don't find them doesn't mean that they're not there. So I think part of this, as we're kind of diving into this too, is you had mentioned limits of detection. So let's kind of help everybody kind of define two key terms around that. So limit of detection and limit of quantification. What do those two things mean, Doug?
Well, an LOD is very much matrix dependent. So what is it are you testing? Is it an ingredient? Is it a food? Is it an environmental specimen? And the minimum amount of the analyte, it could be a chemical, it could be a microbe, it could be a portion of a microbe, let's say a cell wall for an ELISA test or a lateral flow, or the genetic information, whether it's RNA or DNA. Mm-hmm. And so you have to have a minimum level of that in the analytical portion that you're testing to be detected by the method that you're using. And so the more sophisticated the method is, it may or may not be slow, but it will give you a small limited detection, meaning anything above the LOD, the method should detect it. Anything below the LOD, the method may miss it. Mm-hmm. The analyte could be present in the specimen, could be present in the production lot, but the test can't find it because it's at levels below the LOD. Yep. Similarly, a limit of quantification is you want to measure the amount of the analytes. And so that's a quantitative test. It could be an aerobic plate count indicator in microbiology. It could be the level of a vitamin in a chemistry test. And so in that case, not only does the analyte need to be above the LOD, but you also have to have a method that can actually count how much of that analyte is present in the specimen. Right. And the big problem that we see in the industry, it's just not our customers, it's just not the people I work with, it's really across any industry, is if you pull one small portion to test, how do you know that that portion is representative of the entire production lot or the entire facility or the entire batch of an ingredients? And you really can't answer that question until you test a lot of samples. Back to our medical testing example, those ranges are based on a population of humans who had the same tests. It could be, tall people, small people, fat people, skinny people, black people, white people, males, females, etc. And all of those individuals will have a data point, and then you take an average and you take a standard deviation, and that's where they put those demarcations on. This is what we consider a normal range. It doesn't necessarily mean it's a healthy range. It just means that's the normal range for that test. And if you fall outside, either low or higher, then that's supposed to indicate that you may have a problem with whatever that indicator is trying to measure. Right. And so we have the same situation with food testing. Is we do a lot of indicator testing in our business, and we set some limit that says, for quantitative limit, that says if my measurement is above that, that's an out of spec result. That should indicate that there's something wrong with the specimen that we tested, and in return, there's something wrong in the big picture from which that specimen was removed. If you're only doing one test and you do it once a year, like me yesterday, how do I know that that is representative of if I did the same test 6 months from now, would I get the same number? Well, the chances are I won't get the same number, but I should get a number that's pretty close to that. Ideally, it's within that normal range, which indicates that I'm okay.
Well, really, and this is I love this comparison between these two different things, because really your plant, you can think of your plant as you, right? That's the patient. The plant is the patient. That patient is unique. And there's other plants you can compare to, but that plant is unique. And you need to set up your program specific for that plant. Now, back to your test results. Not only did you just get a snapshot at this one test, but that test now is going to vary to what that test would have been for you 10 years ago or 20 years ago, even though you're the same individual. Things change. So in the plant, your plant, like us, is decaying, and the floors are getting worse, and the grout is falling apart, and the ceiling is starting to leak. All these type of things that you need to manage and maintain, just like us, and you need to keep these things up. In that sense, the plant is exactly the same. You can get that baseline. And I think as we dive into these different type of tests, that becomes very important. I think number one, the thing you always need to remember when you're looking at these different things that you need to test is someone is trying to sell you this test, and they have a vested interest in selling you this test. Is that the right test for you? Maybe, maybe not. You need to figure that out. And this is on this topic is there's something that I would always ask when I go out to plants is, let's take ATP testing, for example. What is, and I will ask this in plants, well, what do you consider pass fail for ATP? And the answer, you know where exactly I'm going on this. Well, 30 RLUs is considered pass fail. And then my follow-up question is always, for what? For what surface? For what are you measuring? How do you know? What is your baseline? And I'll get a look back like, right?
Yeah, and I get asked that question all the time. And I know the vendors of these ATP kits get asked that every time they're dealing with a current customer or a lead. And, people want easy. I understand that. They want to be told what the number is. But is the number in a facility that's doing a fermented dairy product going to be the same as a dietary supplement manufacturer who just blends a mix of highly purified chemicals? The answer is absolutely not. And so there are ways in which you can come up with the right number, but asking an expert is just the first place you can start with. And you need to validate whether that number is appropriate in your facility and for all locations in which you are doing ATP testing.
Right. So when you're starting to use ATP testing in your plant, you need to create and determine for all your different sampling sites, number one, what are the correct sampling sites? And then what are the historical values, just like our blood pressure range? What are the historical values for this particular surface and our particular product? And at what point is that sampling done? Are you doing it right after cleaning? Okay, are you using it as a validation for or a verification for your cleaning process? What is the surface that you're testing? Stainless steel is going to be very different than a rubber gasket. And what are those values associated with that? Are you doing rinse water testing with your ATP? What are you then doing with this information once you have it? We see this all the time, and this drives us nuts on the micro side, is the retest. Right? It's like, okay, I got a 35. All right, I'll just retest. Hey, it passed. Cool, 25. All right, that first result must have been wrong. No, how do you know that? How do you know the second one's right? You touched upon here another very important piece of this as well that applies to all the things that we're talking about here today is sampling. When are you taking your sample? On the micro side, if you have a contamination within your process of something growing in your system and you clean and it kind of got missed and you didn't pick it up, we know from a micro standpoint, if you were to pull different samples during that production run, you're going to have a spike at the beginning of higher growth, and then it's going to trail off during the run as it gets diluted as more product goes through that system. Are you picking that up? A lot of times you'll see on the micro for the sample size, and there was a non-fat dry milk manufacturer in Minnesota that got in big trouble for this. They had a massive recall. They were doing salmonella testing on their finished product. Okay, good. But they were pulling like a 25 gram sample for hundreds of thousands of pounds of production. Really, right? And the COA says negative, right? I'm okay, right, Doug?
Yeah. I want to give a real world example that I'm seeing more and more frequently. And I want to first provide some context so that everybody understands my thinking on this. I live in a household with people who have food allergies. And some of these foods that they're allergic to, it can cause anaphylaxis and death. Mm-hmm. Most of the foods, it causes irritation. Okay, so it's not a lethal event. So if this individual is exposed to a teeny tiny amount of the food item or the protein within that food that causes allergy symptoms, they could die. Okay, so personal bias here. Okay, so food manufacturers, if you have regulated allergens, I also want to put in gigantic parentheses there's only 9 regulated allergens in the US. There are perhaps hundreds of different foods and ingredients that an individual could be allergic to beyond the regulated food allergens. So just keep that in mind. Okay. So if you are doing a process change, going from one allergen to a non-allergen, or going from one allergen to another allergen, you have to do a process changeover that involves removal of the first allergen from that first SKU. How do you do that? Well, you design and validate a cleaning protocol to remove those allergens. Okay? How do you determine whether you have a cleaning protocol that works? Well, you validate it through testing. And I've seen people validate their allergen changeover cleaning process using ATP.
Oh my God.
Yeah, as the only test. So if it's in spec, whatever that spec limit is, they say, yes, this equipment is fit for use for the next SKU. All right. Mm-hmm. So we know that ATP measures energy compounds in organic material. If you remove organic material, which would include allergenic proteins, then the ATP levels would fall. So there is some logic to the choice of that as an indicator, but it doesn't measure allergenic proteins. Okay, are there better tests? Yes. I've seen companies validate their cleaning protocol using a test kit for protein. Mm-hmm. Because allergens are proteins. If you can get the protein removed, in other words, below the limit of detection of the kit, then you proclaim that cleaning protocol has been validated. What's the problem with using protein? Well, the problem is some allergens are stickier than other allergens on equipment surfaces. Dairy proteins, for example, one I know you've got a lot of experience with, can be a more challenging protein to remove from surfaces. Mm-hmm. Certainly if you're a dry blender or a dry process, getting every little particle of that prior run out of that equipment is, for most people, it's impossible.
Yeah.
Okay, it literally is impossible. So you are getting it lower, but it's not ever zero. All right, so protein, maybe not the best choice. Okay, there are other kits out there that can actually measure the allergenic protein. Some of these are easy to do, meaning you can do them in the plant. So these would be the little snap tubes that you see. They could be little lateral flow things like we've all done with COVID tests. Okay, so they're more specific. They're actually measuring the allergenic protein, but their limited detection might be higher than other kinds of technologies. Yeah. So again, you're not measuring zero, but you want to make sure you're getting as close to zero as you can.
And just a couple overlays on that as well too, is just like the blood test in allergy, the allergen data is also based upon populations. So the University of Nebraska with FARP and things like that, they will look at in the general population and say, okay, well, for soy, are people allergic to soy? Oh yeah, they are, right? And then they'll try and set up what appears to be the general threshold for people for triggering an anaphylactic reaction to soy. Okay, it tends to be around 8 parts per million or whatever it is that they set the limit at. That doesn't necessarily jive with that test. So you have to keep that in mind too. You'd also mentioned the product matrix. If you have, and I found this when I was investigating in my past, different test kits for allergens, is the density of that matrix and how that, especially with the lateral flow device, if you've got a really thick, viscous type product that doesn't dilute very well, and you're putting it in that lateral flow and it's supposed to run through that system to be able to test and it doesn't do it, now you've got a false negative. And you don't even know. Putting my regulatory hat on, if you work in food safety as well, now you're into label claims. So to your point, Doug, if you have an allergen and you switch to another allergen and you don't clean that allergen, your product is now adulterated. That's an easy call for FDA. That's instant recall. How do you then prove that that is not there? And anytime you switch over like that, it's very, very important to have that correct. And you have the proof that it's correct, because you're going to get challenged on that. Something as well is with certain types of products, there's normal background allergen cross-contamination. So think grain products. Just like your analogy with the plant, sometimes it's very impossible, especially with dry plants, to get everything out. It's the same with trucks transporting grain. One day it's doing wheat, the next day it's doing soy. And yeah, they kind of blow out the truck and they get everything out that they can, but there's always a little bit there. All of this points back to your label claims. If the marketing group wants to put on the label soy-free, gluten-free, dairy-free, what does free mean? Free means 0, not there. It doesn't mean not detected. So you could have a limitation that FARP puts in place for soy of 8 ppms, but your test is sensitive down to your level of detection at 5, and you have a hit. That product is not fit for human consumption. It's adulterated.
Yeah, so the methods that we would do in our labs, because we're reference labs, people want the best number that technology can deliver. So we will do primarily ELISA kits that have a lower detection limit, meaning it's more sensitive, right? Doesn't mean it's poorer, it just means the detection limit number is lower. And I've been into facilities helping them validate an allergen cleaning process. And these facilities might have worked for 2 weeks to try and remove dairy protein from equipment. And they were using some of these easy to use kits and, do-it-yourself kits. And they were very, very confident that they had removed dairy. And I was there on behalf of a brand owner who wanted to process their product on this equipment. And they wanted to make sure that the allergen cleaning process was effective. And so all their data showed, we don't have any more dairy protein. So I went in, they had run a big dairy SKU through, they went through their cleaning process. It took them 12 hours. And then we started taking samples, sent those to the lab. The vast majority of those came back out of spec. Including equipment that had been cleaned out of place. It was shiny. I would lick it in no hesitation. But if I was dairy sensitive, that one lick could have killed me.
Yeah.
And so they got the data back, and not only were they absolutely destroyed because they thought they had it figured out, but the big giant customer they were trying to woo said, we're out.
Now, if in that production facility, you had allergen changeovers independent of this potential customer, now you got a big problem. Now you have determined that your allergen cleaning process is ineffective. Now what do you do? So a lot of times, just like kind of the person who doesn't want to do their blood work because they don't want to know, now you've stepped in a giant heap of something. And now this gets into the legal side of things now too, is, we could potentially have been producing product that was adulterated with allergens and we didn't even know it. So sometimes companies will kind of take, and this is the absolute wrong thing to do, is, all right, if I don't know it, it doesn't hurt me until one day it does, just like your own health. So those allergens and the ATP, setting that base. And so you had mentioned something within these inherent in this that I wanted to make sure everybody paid attention to, is the sample size, but also the sampling methodology. So if for your ATP testing, you're going to get very, very different results if you're taking your swab or your sponge or whatever and just really getting in there and really working it versus this, boop, right? And you're fooling yourself. That sample size that we had talked about earlier, how are you pulling those samples? How much sample are you pulling? One of the things that's very effective with allergen testing is rinse water. Why? Because rinse water is looking not only it's looking at the entire liquid that is circulating through that system once you do that cleaning cycle versus a small square of particular easily accessible, right? It's always easily accessible surface that they always test, just that one surface. It's like, okay, great. Well, we know this one little 5x5 square is clean, but we don't know about anything else. And sampling is very important.
Yeah, I did notice there was a question in the chat about what my opinion or what our opinion was about composite sampling. So you pull random samples, whatever the number is, and then you composite them, and then you do one test on that composite. Okay, so I'll use the analogy here: dilution is the solution to pollution. So if just one of those samples is out of spec, when you commingle it with other specimens in a whatever your composite protocol is, you're going to dilute that analyte perhaps below the limited detection of the method.
Right.
And so you could have an out-of-spec lot that could kill someone, but because of your sample compositing scheme, I mean, composite math does not lie. And anybody who tells you that it does, I want to know what math classes they took in high school and college to come to that conclusion. Okay, so the math doesn't lie. So you could be reacting on an in-spec result for that composite sample, yet your production lot is out of spec.
Right.
So,
and
a lot of people do this. And I hope folks on the call understand what that risk is. Because it is risk, and if your production lot winds up in a recall or harming consumers, and someone looks at your test protocol and they say, hey man, you were negligent with your sampling because you did composite sampling. How do you justify that behavior? And the reason why people do that is because they're trying to save money.
Yes.
Honestly, that is the reason.
Yeah.
And so imagine that is your answer if you're on the witness stand and the plaintiff's attorney asks you, please defend your composite sampling plan, and your answer is, doesn't have to be blatant answer, but if that lawyer is clever and the jury is listening, they will absolutely be able to demonstrate that the reason why you were doing it was to save money.
Yep.
And if you want to have better evidence of negligence, that's way up there on my list of you have just torpedoed your defense.
Absolutely. And so two different examples here from the inbound and outbound side that I think are relevant. So back in my microbiology days in the lab, so I worked for a company that made dairy powders, and we would do class 1 FDA testing, which is a very large sample. So we would do duplicates of 375 grams in 3,750 mils of water. So giant jugs. And we had giant incubators to run this process through. And we were actually doing FDA banned methodology at that time. So the base, plating, bedrock of everything is based upon whether or not it complies with this standard. But what we would do, Doug, is we would get individual production run samples from the plant by pallet, giant boxes of samples. And then we would prepare our own composites in the lab. We didn't let the plants do the composites. And then we would do the composite test, but we would keep all of the pallet samples for that production run. If we had a hit on that production run, we would go back then and begin testing individual pallets with that run. And that wasn't necessarily meant to release product. That was meant to figure out where the problem was coming from within that process.
Yeah, and again, some people are pinging here on, okay, one is, N20 versus N60. And so where those numbers come from, that's just the number of samples to pull
right
for production lot clearance. Where those come from was a paper that was published by the International Commission for the Microbiological Specification for Foods, probably published 30 years ago or more. Okay. And that was based on trying to improve the odds that you could find an out of spec for a pathogen when you're trying to do lot clearance testing.
Mm-hmm.
Okay? And they had 3 categories that were based on risk of the final product. Product that's not going to be consumed in its current state, so it's not considered ready to eat or ready to consume. You could maybe pull 15 samples. Product that is ready to eat, but consumed by the average population, whatever that is, then you would pull 30 samples. If you knew the product could be consumed by a high-risk consumer, infant formula, daycare centers, congregate care facilities, hospitals, then the recommendation would be to pull 60 25-gram samples. And so that's where those numbers come from. Does that, and statistically, N of 60 gives you better odds of finding an out of spec than an N of 15.
And yeah, you want to drive yourself nuts. Just go to, on the web, generic sample size generators based upon individual units, and look at the number of samples you would need to pull for a 95% confidence limit. Right? And then you'll lose your job when you request those samples from your operations group, because nobody's going to be able to do that. It's too huge.
Yeah, I did this yesterday for a client. They had 48,000 packages in a lot.
Uh-huh.
And they were concerned that the lot may be contaminated with mold, because the sample they sent to the laboratory, they found a mold count of 20 per gram. So when you think about that, and their spec limit was less than 10, okay? So that sample was out of spec technically,
Right.
but from a lab's perspective, you know how many colonies were on the plate that was counted? 2.
Mm-hmm.
Now, Mother Nature blows mold spores around all the time. Would not be uncommon to have one of those just randomly fall on a plate and go, well,
Yeah.
you got a count of 10. Okay. So they said, how many samples do we need to test to figure out whether this production lot truly was contaminated? I went to one of those online calculators, and it came up with a number that was about, depending on the risk level you wanted to assume, for the lowest risk, it was 800 samples.
Mm-hmm.
Now, as a laboratory operator, I think that's fantastic. You know, this calculator, this bot just made me a lot of money, because now I'm telling this customer, yeah, you need to send us 800 samples. And to your point, how many in the business are actually going to do that?
Yeah. There used to be, and I don't see this anymore, is to your example of 2 per gram with the mold. We used to put a little disclaimer at the end of that when you put your results. Less than 2 per gram estimate, EST, meaning you are outside of the statistical variability for that test at that dilution. So normally you want colony counts between 25 and 250. And if you're outside of that, now randomness falls into it, because again, bacteria do what they want to do. And I think this is something that's really important, because companies get into hot water, PCA, over this type of thing all the time. Is, oh, we had a hit. Let's do a retest. Good news, Doug. The retest was negative. Can we release the product?
Yeah. So regarding retesting, I think retesting, if you get an out of spec, is not a bad thing to do, simply because your single data point says that sample was out of spec. You really don't know whether the entire production lot was out of spec at that point. This tells you that sample was. And so you could pull more samples, do more testing, because that will help you maybe conceptualize the magnitude of risk that this lot actually has. So again, the analytical methodologies assume that the target that we're going after, whether it's a chemical or a microbe, or a piece of DNA or a piece of protein, is uniformly distributed throughout the entire production lot. And very, very few foods are truly so uniform that one could expect pulling a fewer number of samples might be appropriate. And the one, a couple of examples I might give you would be a tanker full of milk that has stirring paddles in it. So could the analytes be uniformly distributed in that tanker full of milk? Maybe. But finished products, you've got different sorts of things happening earlier in the production run compared to later in the production run. So is every gram of material produced the same? Usually not.
Right. And this circles back to my early commentary, chemistry testing versus micro testing. Chemistry testing, sure, you can expect that all things equal, that the salt level, for that particular product is going to be pretty close to that same baseline, plus or minus, based upon that test. Micro, completely different world. Now, overlay all of this that we're talking about internally onto your suppliers. Every problem that you have internally with everything that we're talking about here, they have that too. And it drives me nuts, Doug, when I hear this. It's like, well, no, I've got a COA. I'm okay. Really? How many times? This all gets back to that factor which we have to live with, which is money. And if you're producing a product to sell for manufacturers, do you want to go out and do all this testing that they're not even really asking for? Probably not. So say, okay, I'm going to test my product once a year, and then I'll use that result for my COAs. Do you understand that? What methodology are they using? Because a lot of times on COAs, you don't even see that. It'll just say Listeria negative. And you're like, what does that mean? Often is it tested? What was the sample size? What methodology you used? To your points, how was this validated and verified that this works? Are you doing this testing internally? To Paul's point earlier, are you using a lab that's 17025 certified that actually is qualified and audited to run the test that they're running? All of these, you need to understand all of these things. People just accept too much at face value.
Yeah. When you look at recalls, particularly the ones last year, a pretty sizable portion of that was due to the brand owner inheriting a problem from a supplier. Mm-hmm. And when you look at FSMA expectations on supplier verification, the one that is pointed out is, has the supplier participated in and successfully passed a food safety auditing scheme? Okay. And so your verification is, show me the proof that you have been audited. What was your audit score or whatever? Okay. Yet, pretty much anybody in the industry, at least in the US, you want to sell your product either to a retailer and to many food service operators, certainly to another manufacturer. This is a cost of doing business, as being part of one of these food safety auditing schemes. So everybody has them. Yeah. The companies that get bitten are the ones that solely rely on that as the verification data point that the supplier is fit to do business with. Yeah. And so I am often asked to referee a relationship between a buyer and a seller. And so the buyer is going to put some sorts of purchase specifications on the seller. Many of those requirements beyond price relates to food safety or food quality measurable attributes. And so, and again, this happens almost every week for me, where the supplier did the verification test, came up with these results. The buyer did their own verification testing, came up with their results, and they're not similar. Who's right and who's wrong?
Right.
That's the question for me. And it's almost impossible to answer that question, because you're dealing with time differences, maybe temperature differences, laboratory differences, method differences, all these interfering variables can change the number that the supplier gets and the number that the buyer gets.
Right.
And so I just counsel folks and just say, okay, let's start with this question. Is the spec limit of the buyer reasonable? So they may have come up with specifications that just are too stringent for the supply chain for that material.
Right.
And so it becomes a conversation with the buyer.
And to Jose's point here, contracts, right? This is why contracts are so important. You need to figure these things out in advance of when this happens, because at that point, now it's just a fight over money. Who's paying for what? Who's paying for the downgrade product? It's your responsibility as the purchaser to make sure that your supplier understands what you're concerned about. So if you're an infant formula manufacturer and you're buying non-fat dry milk, you just can't go out and buy any non-fat dry milk. You need to go out and buy non-fat dry milk for infants. And you need to talk with those suppliers and say, hey, listen, we have a very sensitive customer. We have questions for you. We need to understand your process. We need to understand if you're producing spores in your process. Oh, gee, you may have Clostridium in your process. Maybe a little botulism in there. Infants don't really like that too much. They can't protect themselves against that. What are you guys doing to manage that? How are you testing on these things? You need to understand that. At this protein powder manufacturer I worked for in the past, we had varying specifications internally for degrees of quality, all the way from basic animal feed type products, all the way up to pharmaceutical grade. And there was pricing with each of those different tiers because we internally had different specifications and testing methodologies. That supplier who is supplying you doesn't know your limits and your concerns. When they produce that spec that they give to your buyer, it's the biggest spec that they have. You can drive a truck through that spec. They want to sell you whatever they have. It's your responsibility to limit that spec, get that in writing, and then to Jose's point, make sure that if those limits are then not adhered to, that you have an avenue for recourse.
Yeah, this is an interesting highlight here, and something that you guys want to consider with your food safety culture, and that is how much control does food safety and quality have over purchasing? And many companies, you've got a purchasing manager, they get rewarded by what? Buying the cheapest stuff they can buy to reduce your costs. Food safety is like, well, we need to make absolutely sure. So now you're driving up the cost to buy that lot.
Right.
So, there are many companies where the two don't even talk to each other ever.
Which is a huge mistake. Write this down. This is super important. In these conversations, the only point you have any power is before the contract is signed. That person selling you that raw material wants to get that contract signed and start making money. You need to stipulate before that's signed. If you try and go back after the fact, it's going to cost you money. Oh, well, sure. Yeah, it wasn't in the original contract to test for Clostridium botulinum, but yeah, we can do that for you. It's going to cost an extra $1,000 a lot every time we ship something to you. Now the buyer is going to be not too happy with you. The procurement group and the food safety teams, the quality teams need to be just absolutely hooked together at the hip. Those two groups need to work together. Once that problem comes to the plant and you've accepted it, it's too late. It's too late. You need to prevent it from happening in the first place, just like the health analogy, right? So it's preventive versus reactive. Much better to be in the first group than the second. Great comments here, guys. Love this. Yep. So Cortland, quality agreements must supersede. There we go. The MSA. So we're at the 5-minute mark already, Doug. So we've covered an absolute ton of territory, and I'm sure everybody's heads is swimming around this if you're contemplating this for the first time. What type of thoughts would you like to lead people with here today?
Well, for those of us in food safety and quality, I would say the major objective you should have in your jobs and your careers is to be able to prove beyond a reasonable doubt that the food that you are manufacturing is fit for use. Because that proof beyond a reasonable doubt, number one, it protects you personally from being fingered in a negligence charge. So that's where not only are you going to lose compensatory damages, but you're also potentially going to have to pay for negligence. But it also sends the message to your employer that food safety is a non-negotiable item. And you're not making these requests because you want the company to spend more money. You're making this request because this is a revenue protection activity. Because if you're involved in an outbreak or a recall, the brand damage to that will far, far exceed what you're spending on gathering these validation and verification data points.
Very well said. As I've been talking about more and more here on the chat, risk is contextual. And I think in food safety, this is something we forget. We look at non-fat dry milk and we say, oh, here's the chemical, physical, and microbiological threats associated with non-fat dry milk according to FDA Appendix 1. Done. Wrong. Just like that generic range of lipids for your blood test, that's the general population. That's giving you a starting point. You need to take that material now and say, how does this risk apply to my plants, my processes, what I do, and you mentioned this earlier, Doug, my clientele. Who am I selling to? Because a lot of times, think about your sales force in the company. They don't think about this, right? So if you've got a product, let's say that you're doing a beverage and you're going out and the sales people are doing what they need to do. They're going into Kroger, they're going into Publix and whatever, and they're selling it. They're having fantastic success. Well, they start running out of avenues to sell their products. What's the next thing? Okay, well, let's start selling to hospitals. Let's start selling to school districts. Let's start selling things into the WIC program, etc. Well, your risk profile in the company has now just changed. How do you manage that? And you need to be aware of these things and look at those and figure those out, because these things change all the time. There's a reason why with the regulations, FDA says you need to review your food safety plan at least once a year, baseline, right? But they also throw the caveat on there that says, or when things change. And people forget that second part. And things are changing all the time.
One pitch for attendees is, in your respective lines of business, search out trade associations that have food safety as one of their primary objectives.
Yeah.
And many of these organizations were formed with only one purpose in mind. That was because somebody in that line of business caused a giant outbreak, or even a small outbreak, or a bunch of recalls, and everybody else manufacturing similar products came under the thumb of a regulatory agency, consumer advocacy groups, or their customers, saying, can this happen to the product you're manufacturing? And so they have a lot of resources available to their members. They talk about it all the time. It's a great way for you to get continuing education that is more specific to your line of business than what Brian and I talk about whenever we get together.
Yep. Because that's a very good point, because we have to talk about these things generically. We're talking about a whole lot of different variables here. And the idea here is to point you in the right direction. Now, one of the things, Doug, that I get here in the chat every now and then too is, hey, Brian, the chat last week looked really amazing and I missed that. Can I go back and watch that? And so the answer to that is yes. And I put together a database which includes every single chat for the last 5.5 years that I've been doing. And they're arranged by topic. So if you want to go in and say, man, I missed that conversation that Brian had with Doug 6 months ago. I really want to see that. This database gives you access to all of that. And as people on the chat who are the regulars are aware now so I'm retiring and this chat is ending at the end of March. So April 3rd is going to be the last food safety chat. So there's 4 or 5 chats left, and that's it. But I didn't want this information to be lost, because this, just like these trade associations, this is valuable information that people need to apply. So with this database, I put this together. I'm giving everybody unlimited lifetime access for this database for $10, right? So for less than a trip to Starbucks, you can go and access this data, which includes these videos, MP3 files, summaries, transcripts, notes, like my notes from the show of what the guests and I go over with. And then you can go back and revisit this information and apply that to what you're doing. So hopefully people will take advantage of that.
Well, Brian, it's been a privilege participating in these over the years. And I'm personally going to miss you, but I know how to find you. So thanks for doing these. Really a tremendous resource.
Thank you very much. Yeah, it's kind of like you were saying earlier. Yeah, food safety people, we really don't go away, right? We just kind of fade. We just kind of fade away. So with that, always a pleasure, Doug, to have you on the chat. Just a font of information every time you're on. Thank you, sir, for all that you do for food safety.
Have a good day.
And thank you, everybody, for attending. And of course, for the meantime, we will be back here next week at the same time, 8 a.m. Mountain Time, 10 a.m. Eastern. We'll bring another amazing guest like Doug onto the chat, and we'll have a conversation with you around food safety. But in the meantime, enjoy your weekend. Have a great time, and we'll see you back there then. And keep doing what you're doing. We'll see you then. Bye, everybody.